Welcome AP Bio students, this is Mrs. Rydell. And in this little video, we're going to take a look at, refresh our bio brains about photosynthesis. This is a process you should have learned about in Bio 1. You should have notes about this, and you should have this little drawing that we're going to do in your bio notes, but we're going to kind of go back over it and kind of refresh our brain. And so let's think back to where this is going on. If we're doing photosynthesis, we're using chloroplast. Chloroplast is our photosynthesizing organelle. Double membrane organelle, remember that that one in mitochondria are the two organelles besides the nucleus that have a double membrane. Thylakoid sacs inside look like little stacks of pennies stacked up inside and interconnected between the thylakoids. And then this space surrounding, fluid-filled space surrounding the thylakoid sacs is called the stroma. Remember the stacks, one stack is a granum, plural grana, more than one. And if you look inside the chloroplast, now this is where the enzymes that are doing photosynthesis are going to be found, is in the thylakoid sac membranes and in the stroma area of our chloroplast. So we're going to kind of zoom in on one of these little sacs, one of these little thylakoid sacs. And we're going to draw out the process of photosynthesis. So here's my little sac here, my little thylakoid sac. And we're going to kind of take a peek at what's going on here. I'm going to outline here. Remember now, this is my chloroplast. So this is going on inside of the chloroplast. And remember, multiple thylakoids, not just one, stacked up. But we're going to kind of zoom in on one and look at the enzymes that are involved and look at what's happening. Take a peek at our little thylakoid here and look at the enzymes that are going to be important for this reaction. The first ones are the photosystems. Photosystem here, photosystem here. We have two photosystems. They're complexes made up of lots of different light collecting pigments, chlorophyll, the two forms of chlorophyll, A and B, and those other accessory pigments, the xanthophylls, carotenoids, that are helping the chlorophyll to collect the light energy. And we're going to identify them. If you remember from bio, they're named a little oddly because of how they were discovered. So this photosystem here, photosystem number 1, actually is second in the line. And photosystem number 2, PS2, actually sits in front. And it's because photosystem 1 was discovered first, and then later on we found photosystem 2 and realized in the sequence it's out in front. So the numbers run a little backwards than what you would think. Connecting now those two photosystems are proteins that are part of what we call the electron transport chain. These are proteins that are attached to the thylakoid membrane. And remember when we talked about regulating pathways, enzyme pathways and biochemical pathways, and if you have enzymes, multiple enzymes that are working together to complete a pathway, it's more efficient to attach them together to a surface and put them in the sequence that they're going to be used in that pathway. And that's what is happening here with our photosystems, our electron transporter, our ATP synthase. They're all part of a pathway, and they're connected together in sequence in the thylakoids. If you remember now, this is my thylakoid membrane here. And thylakoids, the thylakoid membrane is made out of multiple sets of these electron transporters, photosystems. It's not just one setup in each thylakoid membrane. So if you look now, let's put some labels on where we are. This is my thylakoid membrane, part of my thylakoid sac. And phospholipids, remember, membranes are made out of phospholipids and proteins. There's my phospholipids. These actually go all the way around. We just didn't really put them in the picture because we want to kind of focus on what the proteins are doing. This here would be my thylakoid space, also called the lumen. And then out here is my stroma. Stroma is this fluid-filled space that's outside of the thylakoid sacs. So remember, we're inside the chloroplast now, inside the chloroplast. So we have our setup, the two photosystems. These electron transporters, let's put a little label on them. They're called electron transport chain this whole series of proteins. I'm going to mark them up here so I remember what ETC stands for. Electron transport chain, sometimes called electron transport system. Okay, and then our last enzyme that's part of this whole process is going to be this one. This is our ATP maker, ATP synthase. ATP synthase is a transport tunnel protein. Remember, in order for ions or some kinds of molecules to get across membranes that can't pass those photic tails, we need to give them a tunnel or a channel. And this is an ion channel. It's an ion tunnel. This is ATP synthase. The name tells you what it does. ACE at the end tells you it's an enzyme. Okay, so these are this is our setup for doing what we call the light-dependent reaction. Remember, light is the part that is the photo part of photosynthesis. This is we're catching the light energy using those pigment molecules. So we're going to start our process by zapping our chlorophyll. Light is going to hit those photosystems. It's going to be collected by the accessory pigments and pass to chlorophyll, and the energy from that light source as electrons. Chlorophyll electrons are going to be charged up and excited by that light energy that hits them, and they're going to be boosted to a higher energy level. As the electrons fall down in energy, lose energy, and fall to lower levels, they are going to be passed down this electron transport chain. So I'm going to put some electrons in here. We're going to follow where they go. So 2 electrons are going to get excited. And remember, electrons have a little negative charge, so I'm going to put E- for electrons. They're going to pass from chlorophyll or from those accessory pigments to the chlorophyll, and then chlorophyll into electron transport chain. We're going to pass down the line, electrons moving as they go, 2 electrons going down the electron transport until they reach photosystem number 1. They've lost energy. Each step of the way, they lose a little bit of power. These molecules that are part of the electron transport chain have another function. In addition to being electron transporters, they are proton pumps. Proton pumps pump hydrogens. So we're going to put some hydrogens into our picture here. I'm going to label hydrogens. We have some hydrogens floating in our cell, free floating. Remember, water dissociates at pH 7 to give us a small amount of hydrogen ions and as hydronium ions and OH ions. So we always have a little bit of hydrogen kind of floating, free floating in our cell, kind of background hydrogen. So those proton pump now are going to pump some of that hydrogen as the electrons move down the chain into my thylakoid space. Not only are they passing electrons, they're proton pumps. Okay, so we're going to pass down the line, pass down the line. When I get down to photosystem number 1, those electrons have lost a lot of their power. They need another little boost of energy. So we're going to hit this photosystem with another zap of light, charge up the electrons again, and give them the energy they need to jump to our last, from photosystem 1 to our last electron transporter. And then finally, from that transporter, jump to our electron carrier here, which is NADP+. NADP+ is an electron carrier. And so we're going to take NADP+, we're going to add those 2 electrons that it receives at the end of the line. This is our final electron acceptor. And notice the difference in charge, 2 minuses, 1 plus. We're going to reduce this molecule by adding a hydrogen ion as well. And I'm going to get a charged up version, which is NADPH. This is the charged up version of NADP+. It's the reduced form. And I'm going to put a little excitement around this molecule. This is a high energy electron carrier, so we're going to give it a little power here, a little juice. Okay, now we have a problem because we zapped our chlorophyll, we passed electrons down the line, and now handed them off to NADP plus to become NADPH. Now I got a problem because the next zap that comes, I've already passed electrons, and there's a little space there where those electrons left, and I need to refill that space with replacement electrons so I can keep this process happening. I can keep collecting light energy. And where those electrons to refill the space are going to come from is they're going to come from water molecules. So if you look at water, and we have water in our cell, remember cytoplasm, our cell, our blood is mostly water-based, so there's water in our cell. I'm going to give it the hydrogen color, because that's where those hydrogens, free-floating hydrogen ions in the cell are coming from splitting water. Hydrogen, and I'm going to make the O be a little different color, because I want to kind of keep track of what happens to my atoms in this process here. So H2O, water. And we're going to kind of color code that. Now, water molecules are going to split apart. And when they do that, I'm going to get an O. And I'm going to get 2 hydrogen ions, H2, 2 hydrogen ions. And I'm going to get 2 electrons. And those 2 electrons are going to pass to chlorophyll and fill in the space, the gap that was left when those original electrons jumped and went down the electron transport slide. So now I've replaced those electrons by splitting a water molecule. And now I'm ready for my next zap that comes. I have electrons to move down the chain again, pass down the chain. We're going to pump hydrogens into my, as they go down the line, pump hydrogen ions into my thylakoid space. And then I'm going to split another water molecule, H plus, H plus. I'm going to get another water molecule split, another O, to replace those electrons. So I zap with the light, jump the electrons, they pass down the electron transport, they're stored in NADPH, and then I replace those with water from the water molecules, and then I'm ready for my next zap. Okay, so what we're doing as we're running the electron transport chain is creating a hydrogen gradient. We're pumping hydrogens into the thylakoid space from the stroma side, and we're also putting hydrogens into that space by splitting water molecules. So hydrogens are coming from 2 places, the proton pumps of electron transport and splitting water to replace the electrons that are lost. So oxygens combine together. Oxygen is a very reactive molecule. It doesn't ever stay by itself. I'm going to combine 2 together. They're going to buddy up and make oxygen, O2, an oxygen gas molecule. That's why we kind of put a little cloud there. And that's one of our products of photosynthesis. Remember, plants give off oxygen as they're providing the oxygen in our atmosphere for us to breathe. So oxygen is going to leave the chloroplast, leave the thylakoids. It's going out to the atmosphere. It leaves the leaf through openings called stomata. And that's how the carbon dioxide, our other gas we're going to use, gets into the plant. But it's going to escape, it's going to leave the building, it's gone, adios. We're losing oxygen as water is split. We're going to lose that oxygen as a waste product. Okay, now I got another problem because I created a hydrogen gradient here by putting all these hydrogen ions into my thylakoid space. And you know that if you create a concentration gradient, high on one side, low on the other, those ions are going to want to move. They're going to want to move back to the other side. They want to go from high to low. That's just plain old simple diffusion, right? We've created a concentration gradient. And if I give them a transport tunnel to move through, those hydrogens will move automatically across from high to low. I don't have to do any power or add energy. Remember, that's a kind of passive transport. We're just going to go from high to low. So hydrogens, if I give them a tunnel, like ATP synthase, which is an ion tunnel, a hydrogen ion tunnel, we're going to move our hydrogen ions from inside the thylakoid space back over to the stroma side. We're going to use the gradient to do that. Okay. And so as I'm moving hydrogens, automatically, no energy required to move them. They go down the gradient. Through that tunnel, the power of moving those hydrogen ions through is going to be like water over a dam. It's going to be able to be collected and used to provide the power to charge up our ATP. So I'm going to take ADP here and add a phosphate group and become ATP. I'm using the power of the hydrogens to create the energy I need to make an ATP molecule. I'm going to add a little power to this molecule. This is a high energy molecule. Remember, that's our rechargeable cell phone battery, and we're charging it up, in this case, using the power from our gradient. That process of moving down a gradient, having molecules move down their gradient to create energy, and in this case making ATP, is called chemiosmosis. And that process of using the hydrogen gradient to make ATP is what's going to store that light energy as chemical energy that now we're going to have some power to do the synthesis part of our photosynthesis. So now take a look at where we are. What we've created now is these energy molecules on the stroma side of our thylakoids. Remember, we're creating the hydrogen coming through, and we're making these energy molecules over in the stroma. And that's where the Calvin cycle, the next part of our process, the light independent part, is going to happen. It's called independent because now we've used the light energy to store up some chemical energy, and I don't really need the light anymore. Originally, these were called the dark reactions, but they kind of thought that we're going to use the light during the daytime and make all this ATP, and then we're going to, at nighttime, the plant can use that ATP to make the sugars. And we know that's not really true. Plants make sugar all the time, as long as there's light to make the ATP. It can go on a little in the dark as long as we have these molecules stored up during the day. But once those are used up, we really can't continue during the dark hours without more light to make more of these energy molecules. So we call this the light independent now, independent part. And this is going to be the synthesis part now of photosynthesis. We're going to make the sugar, and we're going to use the energy we stored in these high energy molecules to do that. So we're going to send our NADPH down to the next part of our cycle. We're going to send it down. We're going to drop off the hydrogens, hydrogen ions, and the electrons. And then once we drop them off, that carrier can come back, the NADP plus comes back to be recharged again by the light reaction again. This is a reusable electron carrier. And same with ATP. We're going to send our ATP down to the cycle. It's going to drop, lose the P, and then provide power. And then once we lose the P, that ADP can come back again and get recharged up again by ATP synthase in the next set of light reactions. So we're going to provide the power to do this, reducing power with our NADPH and electrons, and then ATP is going to provide some more power to do this. And the molecule that we're going to incorporate here is going to be carbon dioxide. So I'm going to put a little C here for CO2. This is our little gas molecule now. And here's a really important idea, big idea in AP. Where does the O that's going to come from, ends up in sugar, come from? And so I want you to look back and see what color you colored your O up here. Remember, these oxygens that are coming off hydrogen have left the building already. They've gone off as oxygen gas. And so they are not going to be part of our sugar molecule. The sugar, the oxygen that ends up in sugar, is coming from the carbon dioxide. So I want you to pick a different color to make your O here. This is not the O from water. This is O from carbon dioxide. We're going to use carbon dioxide now and the energy from these energy molecules we made in the light reaction to do what we call the Calvin cycle, sometimes called Calvin-Benson cycle, from the guys who figured this pathway out, or the light independent reactions. And what ATP says this year is that you do not need to know all the intermediates, so we're going to just put molecules here. There are molecules all through this cycle that receive the carbon dioxide. There are enzymes that do chemical reactions to convert the molecules and do some chemistry and move things around. If you look at the AP syllabus and what it says, it says memorizing the steps in the Calvin cycle, the structure of molecules, the names of enzymes, with the exception of ATP synthase, you should know that enzyme, are beyond the scope of the course and the AP exam, which means we don't have to know these exact molecules, we don't have to know all the enzymes, but we do have to know kind of the overall, what's coming in, what's going out, what's the process. So we're going to incorporate CO2 into the cycle, and as the chemistry happens, we're going to do some rearranging. We're going to kick out sugar, right? Comes out as a 3-carbon originally in the, from the Calvin cycle, and then 2 3-carbons join to make a 6-carbon glucose. And those molecules get recycled, so that's why it's called a cycle. Carbon dioxide comes in, we do some hocus pocus, rearranging, glucose gets kicked out, and those molecules are ready now to accept another carbon dioxide. And what's powering this now is our molecules from the light reaction. So we're going to make a sugar molecule, and we're going to write the chemical formula here for sugar. And I want you to color code this. So it's important to know where these molecules are coming from. The C that ends up in sugar, remember C6, H12O6. The C is coming from carbon dioxide. The hydrogen, C6H12, right? The hydrogen is coming from, where do you think? Look what's donating hydrogens here. This NADPH, the H that it picked up here when it charged up and received those electrons at the end of the electron transport, is going to drop that hydrogen into the Calvin cycle, and that hydrogen is going to be incorporated into the sugar molecule. And then the oxygen, now remember, this oxygen has left the building. It's not coming from the water. This oxygen is coming from the carbon dioxide oxygen. So use your color, keep the color straight. C6H12O6. The carbon and the oxygen from glucose are coming from carbon dioxide. The hydrogens are coming indirectly, or actually directly from the NADPH, but NADPH got them from water molecules splitting. So the H in water actually ends up as part of our sugar molecule. The oxygen and the carbon from the carbon dioxide end up as our sugar molecule. There was actually an experiment done using radioactively labeled carbon dioxide and radioactively labeled water to see where that O from sugar is coming from. That's an important big idea of the O comes from carbon dioxide, not from splitting the water molecule. That takes care of our process, and thanks for listening.